All right, hello. My name is Dr. Matthew Mitchell. I'm Associate Professor here in the Department of Health and Human Performance and also Director of our Human Performance Laboratory. And today I get to share with you some of the neat research we have done in what we call our CHASE study, which is looking at cardiovascular health assessment and risk in college-age individuals. And so before we even get started, I do want to give a shout out. A lot of this research was done, in fact, most of it was done by our undergrads, Nathan McBrayer, Chloe Ramdant, and Sarah Emanuel. Both had parts of this for their undergraduate research projects and also received a PBA QI grant to do this. And so I'm sharing the bulk of the hours that they spent in doing this work. Likewise, our laboratory assistants, I want to give them credit as well. Michaela Golden, John Sutter, Sydney Eppheimer, who did a lot of the grunt work in regards to ultrasound assessment, doing a lot of these fitness assessments over the span of about 4 months. And they did a really, really incredible job. So today what we're looking at is the effect of carotid intimal media layer thickness and differences that we potentially see in college-age students. And so to kind of understand that, I want to give us a little bit of background. Now, it's fairly well known that cardiovascular disease is an incredibly important clinical issue. Globally, it is the #1 cause of death from a health perspective. And we know that there are certain risk factors associated with it. Family history being the most common one. Smoking history, at least recent smoking history. Elevation in LDL cholesterol or triglycerides or alterations in blood glucose. Hypertension, increases in systolic blood pressure above 130 millimeters of mercury or diastolic blood pressure above 90 millimeters of mercury. And we also know that a sedentary lifestyle associated with increased rates of obesity also is a primary risk factor for cardiovascular disease. And so being able to assess these and measure these is incredibly valuable. However, they are just indicators. They are not direct measures of cardiovascular disease. And that's what we attempted to do in the study, is be a little more direct about looking at that. And what we also know is that we can see some of these risk factors, not just in individuals who are 40 or 50 years old. We can actually see these as young as teenagers. There's very good evidence that we see, obviously family history, but elevated LDL cholesterol, elevated triglycerides, alterations in fatty acids, even in teenagers. There's very good evidence. There's elevations in systolic blood pressure and diastolic blood pressure. And we already see this in that young age. And if that's the case, probably not surprising that the chance of cardiovascular event increases greatly later in life. One other aspect that we know, and this has become really important recently, is that we know the incidence of these risk factors are different in different ethnic and cultural backgrounds. We know that certain minority groups, particularly African American, Hispanic, Latino, some Asian groups, have increased rates of diabetes, increased rates of LDL cholesterol, alterations in blood glucose, percent body fat. And so seeing that there are differences in cultural groups is also significant. Obviously, from a healthcare perspective, the cost of healthcare, practically, that's incredibly, incredibly important. And what we also have recent evidence is that there are actually direct measures we're seeing. And the one we looked at in the study, which is carotid intima media layer thickness, which quite literally is the buildup of plaque along the intimal medial layers of the carotid artery and its inflammation in that artery, suggesting that if that inflammation is occurring there, it's likely occurring in other large vessels and in the heart as well. And there's now a good evidence that alterations in this, which is a primary indicator of cardiovascular disease, we do see differences in these values in different cultural groups. And so for this study, we actually had kind of two focuses. One, we wanted to see, can we see some of these risk factors in a college-age population, roughly 19, 20 years old, where we wouldn't necessarily expect that, but maybe we might. And then secondly, can we see in this group direct measures, specifically carotid intima media layer thickness changes? And on top of that, are there differences based on cultural or ethnic background? And that was kind of the purpose of our study. Let's look at our methods. This is a really comprehensive study. We looked at over 200 subjects. It took us about 4 months to collect all this data, which is why it was very labor intensive. You can see on the screen the age of this group and gender, pretty much of what we expect from a college age population. And we looked at a number of variables. First, on every subject, we did a health and fitness assessment, which include not just height and weight. We measured percent body fat using a 3-skin-fold method. We also used bioelectrical impedance to verify those results as well. We measured systolic and diastolic resting blood pressure. We measured resting heart rate, actually using a portable EKG device, which I'll talk about in a second. And then we measured some other variables, bicep strength, which we considered a measure for overall muscle strength. And then through a step test, we estimated their aerobic capacity, or what we would say their fitness level. And we call this the estimated VO2 max. We also did a 1-lead or 3-lead EKG. Now, we didn't analyze this data as in-depthly. Clinically, we typically would want to look at a 12-lead EKG. But we use this just to see are there any alterations in PR intervals, T-wave inversion, ST segment depression, things like that. Would we find that in this group as well? Now, on top of that, and what was more the labor-intensive part of this, is we wanted to look specifically at the carotid artery and some of the unique things going on in the vessel. And so using ultrasound technology, particularly our Accusound Sequoia 512 ultrasound, using a 13 megahertz linear transducer, we were able to look at a really clean image of the carotid artery and not only measure its diameter, we could actually measure the thickness of this intima media layer. On top of that, using pulse wave measures, we can measure systolic pulse wave velocity, and diastolic pulse wave velocity, and arterial stiffness. All of these are also at least somewhat direct indicators of potential cardiovascular risk. And to give you an idea just sort of what this looks like on the screen here, you can see just a picture, and this is actually from our study, a carotid vessel, and starting from superficial to deep, we have skin, then we have some fat tissue, the jugular vein, and then right below that, that's kind of bigger, is our carotid artery. And we actually have technology where we can actually measure this carotid artery thickness literally to the 0.1, actually 0.001 millimeter. And that was the measure that we're really interested in seeing. Is there a difference there specifically in that measure? All right. We also, just to get a little background information, gave all our subjects a questionnaire. And this was simply a family history health survey questionnaire. It had 17 items, which basically addressed information like family history in regarding to cardiovascular disease, diabetes, even personal history regarding health behavior habits, smoking, alcohol use, things like that. Now, obviously, we relied on the honesty of our subjects. We didn't know the subject's specific identity. So there's an amount we can perhaps debate about the data, but it at least gave us some understanding of family history and current behaviors of our subjects. Okay. And then with our data, we did a few statistical analyses. We looked at Pearson correlations to assess correlations between variables. We did a one-way ANOVA with every dependent variable, and where there are significant differences, did specific post hoc tests to find those differences. And then with our non-parametric data, our survey data, we did a Kruskal-Wallis test, and then to assess differences between groups. And when there were differences between groups, cultural groups, we used a Dunn post hoc test to really assess where those differences were. Okay, so now for the fun part. What exactly did we find? Well, when it comes to just the overall numbers, heart rate, blood pressure, things like that, the data we saw was that this group really wasn't very different than the average 19, 20, 21-year-old. We've done these tests in our lab for numbers of years, and so this group wasn't terribly different in regards to many of those values. And in fact, when we look at family history, there actually weren't many differences between cultural groups, whether it's African American or Hispanic, Latino. We were even able to separate these groups into Caribbean descent as well. There were not significant differences in family history of cardiovascular disease, hypertension, even current health behaviors. The only significant difference we found was when it came to diabetes, that there was some significance, particularly in African American and mixed race individuals compared to Caucasian. There was significant increases in the rates of family history, both on the paternal and the maternal side in regards to diabetes. Otherwise, though, that was the only value we really found significant as far as family history goes. What was probably, though, the most important finding for us, and the one that we were really focused on, is looking at this carotid intimal medial layer thickness. And what we found was that there actually were significant differences between some cultural groups. And on this graph, we actually separated this out to about 8 or 9 groups. And what we found is there were some differences, particularly between Caucasian, African American, Caribbean, and Hispanic descent. There were some significant differences. Now, the sample sizes were not equal in these, given our population, but this does potentially lead to a question of why and what is it that's causing these differences. Is it family history? It is other things, and we'll try to address those in our discussion. But definitely one of our most interesting findings was seeing this difference. And I'll be honest, one for us in the lab, we did not expect. We really, as much as we were thinking this may be different, looking at the group of subjects we were using, most, at least from the outside, seemed fairly healthy. To see this difference in the carotid artery was a bit surprising. And what we also found was that there really weren't differences in other dynamics of pulse wave velocities, whether it's systolic, peak systolic, end diastolic velocity, or stiffness index, which just gives us an idea of how the stiff or hardened the artery is. We didn't find any differences between cultural groups, which wasn't surprising, although given that we did find differences in the carotid artery intima media layer thickness, maybe a little surprising that we found differences in one area but not another. Okay, so what did we find? Without a doubt, the most interesting thing was that we did find changes in carotid intima media layer thickness depending on certain cultural groups. And this has been found in other studies, but honestly not many in this young of an age group. And that was by far one of the most interesting findings we saw. But with that, though, there were not any really other significant indicators of cardiovascular disease outside of family history of diabetes, whether it's current personal or behavioral habits of those subjects or other family history. There was not much significance. So let's talk about the data and what does this mean? And that's kind of the thing we're tackling now is what does this mean? And so first, understanding our sample sizes were not perfect. We made a great effort to keep the n's equal in each group, but that was very hard. And so in our Caucasian group, we had a far larger n than in some of the other groups. And that definitely affected some of our analysis, and moving forward, something we'd like to rectify when we continue doing this research. Truth is, we also understand that our study sample is a bit biased in the sense that we did pick from a college-age population. Generally, college-age students are at least more middle class, economically, socioeconomically, and a little more educated. And so understanding of health behaviors and disease is more known in this group than perhaps a normal 19 or 20-year-old. And so perhaps if we looked at a far broader demographic, socioeconomically and educationally, we might see some differences there. And that's hard to deny given the sample that we had. And then the accuracy of the data, and that is something that with survey data we always question, is how accurate our students were. I will tell you, we definitely had a couple of subjects tell us anecdotally, I don't know about my mom and dad. And that was always a challenge in getting accurate data regarding family history. And for that, even identifying ethnic groups obviously was difficult. We did not do DNA testing. We did not go that in depth. We really just went on the subject's personal identification of ethnic or cultural background. And not surprisingly, there could be some fluidity, we'll say, to that. And that's just part of the study that we have. It's part of just analyzing the data. And truthfully, most studies in this area have reported cultural and ethnic background in a very similar fashion. So we chose that direction in a similar light. But I do think the findings here are interesting, and we're looking forward to the next step of trying to answer, okay, so what is it that's different in this group? One thing we wish we had done, and as hopefully our next step, is to look at actual blood chemistry. Are LDLs different? Are blood cholesterol, HDL, even some other more specific enzymes, creatine kinase, things like that, are they different? That may be signifying, yeah, there are actually not just changes on the carotid artery, but maybe actually some things going on at a biochemistry level that are different in certain ethnic groups in this group. So that's sort of our next step in trying to understand some of this data. And then we have some of our resources here. And so thank you. We really appreciate you hearing our data and us being able to share some of our findings with you. And we appreciate the opportunity to share this. Thank you.